Hello, everyone. This video is going to look at photosynthesis, which of course is the process by which light energy is converted into chemical energy, typically stored in the bonds of sugars. And of course, this turns out to be a very important biological process. And as we're going to see, it shares some properties in common with cellular respiration. And we'll kind of look at this in some more detail. First thing I want to talk about is really this difference between autotrophs and heterotrophs. And so when we're talking about autotrophs, we're talking about organisms that are capable of making their own food. And often when we're talking about autotrophs, we are discussing photosynthetic organisms. However, there are some autotrophs that can use chemical energy as well. Specifically, when we're talking about photosynthetic autotrophs, we're talking about things that we call photoautotrophs. So these are things like land plants, like these ferns, or things like algae, which is not considered a land plant, but still does photosynthesis nonetheless, or even things like cyanobacteria. All of these things are capable of creating their own food via the process of photosynthesis. In contrast to these photoautotrophs, or autotrophs, there are these things called heterotrophs. And heterotrophs are organisms that must consume other organisms in order to get energy. So these are things typically that are either consuming plants, like herbivores, for example, or predators that consume other herbivores, or rather predators that consume herbivores, or even predators that consume other predators. Those are all examples of heterotrophs. So most animals, almost all animals, we think of as being heterotrophs, and pretty much all photosynthetic life we think of as being autotrophic, or they are autotrophs. And an important fact to recognize when we're discussing the relationship between autotrophs and heterotrophs is that most of the energy within ecosystems, most of the energy flowing through food chains and ecosystems really is traced back to photosynthesis. That is where the energy is coming from. It's really coming from the process of light energy being converted into chemical energy held in the chemical bonds of sugars. And so this cartoon on the right shows an energy pyramid where we have these so-called producers, and most of these producers, if not all of them, are photoautotrophs, things like plants that are making sugar via photosynthesis. And then that energy is being consumed by things like herbivores that we would call primary consumers. So things like these rabbits here are eating these plants and they're getting their energy from these plants. And then we have things like these secondary consumers, these snakes here, which are getting their energy by consuming these rabbits. And then things like these tertiary consumers that are sort of at the top of the pyramid, like this raptor here that is consuming these snakes in order to get energy. And so the point here is that all the energy that's working its way up through this energy pyramid really has its origins in photosynthesis. And that energy is being transferred up this pyramid from one sort of level of consumer to the next. So autotrophs really do form the basis of ecosystems and provide the energy source that really fuels an entire sort of so-called food web or energy pyramid. Now, we can talk about how the distribution of Earth's major biomes is really actually pretty significantly influenced by photosynthetic life. This obviously is a sort of image of our Earth's globe, and there are a couple things going on here. We can look at concentrations of chlorophyll in the ocean, as shown here on this sort of scale on the left. And so we can look at the ocean and see the areas that tend to have bright green are areas where there's a lot of photosynthesis going on in the ocean. The areas where there's more dark blue, not so much. And then if we look at the continental land masses, we can see something here known as the normalized difference land vegetation index. Basically, this is an index of really low quantities of vegetation going to relatively high quantities of vegetation as we go from these sort of brownish colors to these more greenish colors. And it's fairly apparent looking at this map of our Earth's globe that there are certain areas with really, really high amounts of plant life, like lots of vegetation, high vegetation density, and certain parts of the globe where there's not a whole lot of vegetation density at all. And some of the areas that don't have a whole lot of vegetation density are typically more arid regions, things we think of as being more like deserts, such as the Saharan desert here, or the Arabian desert here, or some of the deserts of Asia here, or some of the deserts of Western North America here, or the deserts of Australia. But then we can see other areas like in the tropics here, in the tropics of South America, the tropics of Africa, the tropics of Southeast Asia, where there's a very high amount of vegetation. Likewise, in the more temperate northern latitudes, the boreal regions are very densely forested. And so these are areas where we see a lot of photosynthetic activity happening, a lot of productivity happening compared to these areas where there's not so much. And so the point is that the distribution of a lot of major ecosystems, biomes, is really all sort of being dictated by really just the presence of where there are more photoautotrophs compared to areas where there are not so many photoautotrophs. And so, what are photoautotrophs doing? They're doing photosynthesis. And really, the main structures of photosynthesis are fairly simple when taken at face value. We have plants, for example, this tree here, and these plants will take up water, usually from the soil, from their roots. And these plants will also take in carbon dioxide. And this carbon dioxide will react with the water, and via a catalyst that we call sunlight, which comes in from the atmosphere. We have incipient solar radiation, I should say incident solar radiation coming in and allowing a chemical reaction to ensue that converts this carbon dioxide and water into sugars, which are generally present in the leaves of this tree, and oxygen, which basically is expelled from the tree and evaporates into the atmosphere. And so this is basically these are basically the key properties of photosynthesis and a very basic chemical equation that describes the sort of process by which photosynthesis proceeds. Now, much like we saw with cellular respiration, this is an accurate chemical equation for the process of photosynthesis, but behind the scenes, it actually is a lot more complicated. There are a bunch of little steps going on along the way that allow carbon dioxide and water to be the reactants that can eventually be catalyzed to form sugar and oxygen. So despite the fact that we can represent the process of photosynthesis with a relatively simple chemical equation, there are a lot of very intricate steps happening along the way in order for this reaction to proceed. It is, like respiration, a sort of complicated metabolic pathway or a series of complicated metabolic pathways that allow this to work. Most land plants, so we're talking about things like mosses, ferns, gymnosperms, and angiosperms. Most land plants are conducting photosynthesis within leaf tissue. And so here is an image of a typical leaf. If we were to look very closely at this leaf at sort of the microscopic level, we would see that this leaf has relatively intricate structure. There are upper and lower epidermis that sort of protect this leaf. And within those epidermal tissues, we have something called mesophyll. And within mesophyll, we have a whole bunch of different cells that make up that tissue. And this is a typical plant cell here. And what is it that makes plant cells unique? Well, plant cells have chloroplasts. And so here is a magnified view of a chloroplast within this plant cell, within the mesophyll, within this leaf. And if we were to look inside of a chloroplast, we would see some even more intricate structures, as are illustrated with this cartoon here. And with these intricate structures, the first thing I'd point out is something called thylakoids. And thylakoids contain things called chlorophyll. And so chlorophyll is this very special pigment that is capable of taking sunlight energy and allowing that energy to basically be converted into organic molecules that can be used to make sugar. And so basically, not all leaf cells are carrying out photosynthesis necessarily, but typically we do see that chloroplasts are going to be most active or usually going to be occurring or most frequently within this mesophyll layer. And that is the place where photosynthesis is generally going to be taking place. Now, we could talk about these chloroplasts in a little bit more detail. As I mentioned, they're composed of these things called thylakoids. And within the thylakoids are where these pigments chlorophyll are found. And as it turns out, these thylakoids tend to be stacked up on one another, on top of one another in these sort of cylindrical type structures. And these structures are referred to as grana. And there are several stacks of these thylakoids. There are several grana that are distributed within a chloroplast. And within the sort of matrix of a chloroplast is something called a stroma. It's kind of like analogous to a mitochondrial matrix, or maybe kind of analogous to cytoplasm within the cell. But this is a sort of freely moving matrix within the chloroplast, and it turns out to be a place where some of the photosynthesis reactions are also taking place. And we're going to talk about that in a little bit more detail. But generally speaking, the structure of a chloroplast isn't crazy complicated. There is a double membrane, kind of like we see in mitochondria or in a nuclear envelope. And within that double membrane, we do find these stacks of thylakoids, which is where photosynthesis is going to happen. Now, what's going on with light? Well, can't have photosynthesis happen without light. So autotrophs are basically capturing light energy from the sun. Here's an image of the sun. Here's some grasses that are probably trying to capture some of that energy. And as I mentioned, these photoautotrophs will take this light energy and use it to make sugar, and will store that light energy in the chemical bonds of sugar, which then can be used for cellular respiration. And what's up with the light? Well, light, as we know it, travels from the sun in energy waves. And these energy waves are technically known as photons. And photons, if you could think of this, are basically little tiny packets of light that are being shot, or rather being emitted from the sun in waves. And this image here sort of shows what one of these waves look like. You've probably seen an image of a wave before. These light waves act just like any wave that we know of, just like waves in the ocean. They have a certain wavelength with troughs and crests. And basically, the length of the wave kind of dictates the amount of energy that the wave can carry. Long waves, that is, waves with really large distances between the crests and just large distances between the troughs, tend to carry less energy than short waves with short distances between the crests and short distances between the troughs. We can also talk about the amplitude of a wave. So the distance between a trough and a crest in this direction is the amplitude of a wave. But mainly it's the wavelength that we're thinking of here that represents the amount of energy that a wave can carry. Now, light waves are really special because they exist along the electromagnetic spectrum. And the sun emits energy in the form of something that we call electromagnetic radiation. And you may have seen this before. But this scale here on the bottom basically shows the entire electromagnetic spectrum, going from very long waves that don't carry a whole lot of energy to really short waves that carry a lot of energy. So these short waves here on the, I'm sorry, these long waves here on the right, we might think of as things like radio waves, like AM and FM radio waves that travel relatively long distances, but don't carry a whole lot of energy. And as these waves start to get shorter and shorter, we get into the realm of visible light. And if you notice, visible light makes up a pretty small proportion of the electromagnetic spectrum. This visible light is typically what we think of as white light. And this white light can be broken down into basically the range of visible colors, going from sort of long wave colors to sort of short wave colors. So if you didn't know this, the reddish and orangish colors tend to have a longer wavelength than sort of more purplish colors. And beyond the realm of visible light, we have what we call infrared over here, and we have ultraviolet radiation over here. You've probably heard of these things. So infrared is sort of relatively long wave radiation that's just beyond the realm of visible light. And UV likewise is relatively short wave radiation that's also sort of just beyond the realm of visible light. And as we move towards shorter and shorter wavelength radiation, we start to get into things like X-rays or things like gamma rays. And as you might know, when we start talking about UV rays or X rays or gamma rays, this kind of shortwave radiation actually can be somewhat harmful because these waves carry a lot of energy and they're capable of damaging DNA. So you might be wondering, oh, why is it that ultraviolet radiation is sort of bad to be exposed to? It can give you a sunburn. It's because it's relatively shortwave radiation that carries a relatively high amount of energy. Likewise, X-rays, if you go to get an X-ray, the radiologist is going to try to protect you from those X-rays, particularly your reproductive cells, because those X-rays can damage the DNA in your reproductive cells. And of course, things like gamma rays, which are associated with like the radiation emitted from things like radioactive elements such as uranium, carry very, very high energy shortwave radiation that can be very, very harmful to you. And so anyway, this is the electromagnetic spectrum. And why are we talking about this? Because this is where light comes from. And photosynthetic organisms are harnessing the energy in these light waves and using that energy in order to perform photosynthesis. And again, we'll talk about that in a bit more detail in a moment. Now, there are basically three main photosynthetic pigments, and these pigments basically exist across something we call the absorption spectrum. And we can identify pigment types according to the pattern of wavelengths that they are capable of absorbing from visible light. So here on the bottom, we see our visible light spectrum going again from relatively, let's say, relatively short wave radiation to relatively long wave radiation. In this case, we're measuring it in nanometers. So this is the wavelength of those different light waves measured in very, very small units, going from 400 nanometers to 700 nanometers. And what we can see is the absorption spectrum of each of these three major pigments. Chlorophyll, which you've probably heard of, I already mentioned it. There actually are two kinds of chlorophyll, chlorophyll A and chlorophyll B. And then these other pigments called carotenoids, which you may or may not have heard of. But what I invite you to look at when we look at this graph is not only what the wavelengths are on this sort of x-axis, but the amount of light being absorbed by these pigments, relatively speaking. And what we notice is that, for example, chlorophyll A, if we look at chlorophyll A's absorption spectrum, oops, we will see that chlorophyll A is absorbing a lot of light, sort of here that is at a low wavelength, relatively low wavelength, and a lot of light that's sort of over here at relatively high wavelength. But it's not absorbing very much light in this sort of intermediate area here where the wavelengths of light tend to produce colors that are relatively green. And this is why photosynthetic pigments that photosynthetic organisms that contain chlorophyll A tend to appear green is because they are reflecting green light. And so actually this pigment, chlorophyll A, is not absorbing green light, it's reflecting it. And we can say the same thing about chlorophyll B. So chlorophyll B tends to be absorbing a lot of light here at the relatively low end of the spectrum and absorbing quite a bit of light here at the high end of the spectrum. But just like chlorophyll A, chlorophyll B is reflecting a lot of green light. Thus, photosynthetic organisms that have chlorophyll B are going to appear relatively green in color. And then carotenoids are mostly absorbing light around this range here, sort of in the shorter wavelength part of the spectrum, and reflecting a lot of light here in sort of the long wave radiation part of the spectrum. And this is why carotenoids tend to have more of a yellowish, orangish appearance. Um, so that kind of explains why carotenoids have that color. For example, maybe this could explain why when certain leaves turn color in the fall, they sort of ditch their chlorophyll, but they still have carotenoids. So green leaves turn orange in the fall because the pigment that you're seeing is carotenoid pigment that is reflecting orange light. And then we might start asking questions about, well, why are there different pigments? And the answer might not be super clear, but one explanation could be that if plants could have multiple photosynthetic pigments, it might allow them to absorb different wavelengths of light. And different plants in different environments might be exposed to relatively different wavelengths of light. And so if they have the ability to be able to absorb these different wavelengths depending on the circumstances, it gives them a better chance of sort of continually being able to do photosynthesis as the availability of light changes. So, for example, as this image here on the right shows, a lot of plants on the forest floor might not have direct access to light, and so the wavelengths of light that they're responding to or that they're able to absorb might not be the same as the wavelengths of light that treetops way up at the top of the canopy are able to absorb. And so this affords a strategy for different plants in different parts of the environment to still be able to do photosynthesis despite the fact that varying wavelengths of light are available for them to do so. So in short, we basically have these three major pigments, and they exist across three independent different absorption spectrums. Chlorophyll A and chlorophyll B have pretty similar absorption spectrums, but not identical. And of course, carotenoids are a little bit different. But the thing to take home here is that we have variation in the wavelengths of light, going from relatively short wavelength to relatively long wavelength, and that these chlorophyll pigments are really absorbing wavelengths of light at the extremes of this spectrum and reflecting mostly green light, and this is what makes most photosynthetic plants green. So what about the actual chemical process of photosynthesis? Well, it's a bit complicated, and we don't have to go into necessarily all the nitty-gritty details, but I think we should cover enough that you understand what's at least going on at the most basic level. So basically, photosynthesis is happening in two major steps. The first major step is something we call the light-dependent reactions. And the second major step is something we call the Calvin cycle, which I think sometimes is referred to as the dark reaction. But let's just think about light-dependent reactions and the Calvin cycle. And these two major steps allow the process of photosynthesis to proceed. So let's start by talking about the light-dependent reactions because this is where photosynthesis begins. And we're going to look at this happening inside of the thylakoid membrane. And just for reference, recall that the thylakoid membrane is inside of these thylakoids, which are stacked up, which are inside of a chloroplast. And inside of the thylakoid membrane are these things we call photosystems. So this here is a photosystem represented in green. This particular photosystem is called photosystem 2. And within that photosystem are the chlorophyll pigments. These are single molecules of chlorophyll, each one represented by a little green circle in this particular image here. And basically what's going to happen is that first, light energy will be absorbed by a chlorophyll molecule inside of this photosystem. And that energy will be passed along from one chlorophyll molecule to the next, to the next, to the next, until that energy culminates inside this thing we call the reaction center, which is represented by this sort of dark green rectangle inside the photosystem. And inside of the reaction center, energy is going to excite one of the electrons of a chlorophyll molecule enough to send it to an electron acceptor. And so in essence, this molecule of chlorophyll has lost an electron to this primary electron acceptor. And then in order for this photosystem, particularly the reaction center, to replace this so-called donated electron, a molecule of water has to be split. And when this molecule of water is split, it donates an electron to the reaction center in order to replace the one that was lost, that was donated to the electron acceptor. And we'll see what the fate of that electron is in a moment, but know that this molecule of water has to be split in order to donate this electron. And in the process of that water molecule being split, oxygen is produced. And so if we go back to our equation here, we can kind of see part of this chemical equation in action here, where we go from having this molecule of water that is sort of transformed into this molecule of oxygen, or molecules of water transformed into molecules of oxygen, ultimately speaking. So that's kind of what's going on here during the light-dependent reaction. Now, what is the fate of this electron that is donated to the primary electron acceptor? That electron is fed into an electron transport chain, which is not terribly different than the electron transport chain we saw during the process of cellular respiration. And so essentially from photosystem 2, again, light is entering photosystem 2, being passed from one chlorophyll molecule to the next, entering the reaction center, exciting an electron, sending it off to an electron acceptor. A molecule of water is split and oxygen is produced in order to donate an electron to replace the one that was um, sent along this electron transport chain. And as this electron's traveling through the transport chain, it's being used to pump hydrogen ions against their concentration gradient into the thylakoid space. And that electron will eventually find its way to photosystem 1, which, much like photosystem 2, will receive some light, and that light energy will allow this electron to be passed on to some other enzymes that will produce NADPH, which, as you might recall, is a very high energy carrying molecule. And this NADPH will be used later for other purposes. And then ultimately, what's going to happen is that as all of this hydrogen is being accumulated within the thylakoid space, very much like cellular respiration, it's going to be used to drive a proton motive force that allows ATP synthase to effectively catalyze a reaction that converts ADP, an inorganic phosphate, into ATP. And if you recall, ADP is adenosine diphosphate, which has two phosphate groups. And with the energy supplied by ATP synthase, a second, or rather a third phosphate group will be added to ADP to create ATP, adenosine triphosphate. And as you recall, it takes energy to add a third phosphate group to ADP. So in the process of this sort of whole light-dependent reaction, there ultimately is an electron transport chain that is used to produce NADPH and ATP. Now, this NADPH and ATP will become available within the stroma, which, if you recall, is sort of this matrix within the chloroplast. So here is the stroma here, and here are the thylakoids. And basically, this ATP and NADPH is going to be fed into the second step of photosynthesis, which is called the Calvin cycle. And so essentially, the light-dependent reaction is harnessing energy from the sun, really in order just to produce this ATP and this NADPH, which are both very high energy carrying molecules. And as we're going to see, these high energy carrying molecules are going to be imperative in the process of allowing carbon dioxide to be fixed into into an organic form that ultimately will be used to create sugar or carbohydrates. And so basically, we can think about the light-dependent reactions happening in these thylakoids, specifically in these thylakoid membranes. And then the product of those reactions are these high-energy carrying molecules that are fed into this other sort of biochemical pathway, this other metabolic pathway that's going to occur within the stroma of the chloroplast. So what about the Calvin cycle? Well, the Calvin cycle basically has three stages. The first stage of the Calvin cycle involves three molecules of carbon dioxide entering the cycle and reacting with something called RuBP. And there is a very special enzyme called Rubisco that catalyzes a reaction between RuBP and carbon dioxide and forms an organic carbon-based molecule from carbon dioxide. And that molecule is called 3-PGA. And so we call this carbon fixation. Inorganic carbon dioxide is being fixed basically by Rubisco into organic 3-PGA. And then through a couple kind of complicated steps that are going on behind the scenes here, ATP energy and NADPH energy are being used to convert 3-PGA, really reducing 3-PGA, so giving it more of a negative charge, reducing it into something called GP3. And then these GP3 molecules are really going to be important for two purposes downstream here in this cycle. One purpose is to create sugars. So these GP3 molecules are 3-carbon molecules, and so ultimately they will be used to synthesize things like glucose and some other carbohydrates as well. But think of GP3 as being sort of shot out of the carbon cycle, out of the Calvin cycle, and used to produce or used to synthesize glucose, which ultimately is one of the end products of photosynthesis. But some of that GP3 still remains in the cycle. Not all of it is used to make sugar. And the reason why is because some of that GP3 is needed to regenerate RuBP in order for this cycle to continue. So some of that GP3 continues along this cycle, and some ATP energy is used to convert that GP3 back into RuBP so that Rubisco can again catalyze a reaction between RuBP and carbon dioxide and allow 3-PGA to be formed again, and this cycle can go around and around and around, and carbon dioxide can continually be used to make sugar. So again, this is a little bit of a oversimplification because there are some kind of complicated steps behind the scenes going on that are not shown in this image. That's okay if you don't see that here. The important thing to realize is that Rubisco is this very important enzyme that's reacting with RuBP and carbon, or that is catalyzing a reaction between RuBP and carbon dioxide to form 3-PGA, which is a 3-carbon molecule, which in turn is reduced by energy from ATP and NADPH to form GP3, yet another 3-carbon molecule. Where did the ATP and the NADPH come from that supply the energy for this reduction? They came from the light reaction inside of these thylakoids. And so the light reaction is fed into the Calvin cycle in order to effectively reduce 3-PGA into GP3, which is used to make sugar. So this is kind of really what's going on with the Calvin cycle. And I invite you to think about this for a moment. If glucose has 6 carbons and carbon dioxide has 1 carbon, how many turns of the Calvin cycle would it take to produce 1 molecule of glucose? That's right. It would take 6 because there's only 1 molecule of carbon in carbon dioxide. So 6 turns of the cycle is required to produce 1 molecule of glucose. Now that might sound slightly confusing because this image shows 3 molecules of carbon dioxide coming into the Calvin cycle. Well, the reason why it shows that is because it takes 3 turns of the cycle, or 3 molecules of carbon dioxide, to produce 1 molecule of GP3 as an output, because GP3 is a 3-carbon sugar. In order to get a 6-carbon sugar, we need to have 6 molecules of carbon dioxide come into this cycle. So think of this as being 1 molecule of carbon dioxide per each run of the cycle. 6 runs of the cycle give us enough carbon to put together a 6-carbon sugar, such as glucose. So this is our sort of basic model of how photosynthesis works in its 2 major steps, the light-dependent reactions and the Calvin cycle. Again, to reiterate, these light-dependent reactions are taking light energy in order to drive an electron transport chain, which requires the splitting of a water molecule and creates oxygen in the process. And that electron transport chain is basically driving a proton motive force, which creates ATP, and also allowing the formation of NADPH. So essentially, we could think of photosystem 2 as driving the electron transport chain that allows hydrogen to come across its concentration gradient, or rather against its concentration gradient, to drive ATP synthase, to drive that proton motive force. And photosystem 1 really driving the formation, the catalysis of NADPH. So again, the light-dependent reactions main purpose is to generate high energy carrying molecules, ATP and NADPH, which are then fed into the stroma out of the thylakoids and fed into the Calvin cycle and used as energy sources that allow carbon dioxide to be fixed into 3-PGA, which is then reduced into G3P, which is used to make sugar. And then G3P is recycled back into RuBP in order to allow the Calvin cycle to continue. And so we can think about our chemical equation. Where do these products and reactants come from? Well, we see that water is being used in this particular step here. The water molecule is being split, and that water molecule is effectively being converted into oxygen. And we can see that during the Calvin cycle, During the Calvin cycle, we can see that carbon dioxide is a reactant that's entering this cycle, and sugar is ultimately a byproduct of this cycle. So we can see our reactants and our products, both in the light-dependent reactions and in the Calvin cycle. It's just that there are a lot of other complicated things going on behind the scenes that are not evident in this chemical equation. But if you study this carefully, you'll see that each of these components certainly are present in the process of photosynthesis from the beginning until the end. So we can also talk about some alternative photosynthetic pathways that plants have evolved in addition to the pathway that we just saw, which is sort of the normal pathway, which is known as the C3 pathway, or the pathway used by C3 plants, called C3 because it basically relies upon the production and reduction of three carbon compounds in order to produce sugar. And these two alternative pathways are C4 photosynthesis and crassulacean acid metabolism, also known as CAM photosynthesis. And these two alternative pathways seem to have really evolved because they afford the plants that use them the chance to prevent water loss. So in some environments, water stress isn't really a big issue at all because the atmosphere is relatively humid or the concentration of water vapor in the atmosphere is relatively high. And so plants aren't really stressed out for water so much. These also might be environments that receive a fair amount of rainfall. So the C3 pathway is perfectly fine in those kinds of environments. But in environments that are kind of relatively hot and dry, where plants might start to become stressed by the availability of water, they might switch to using things like C4 or CAM. And why is water important for plants? Well, we know from what's going on during the light reaction that a molecule of water has to be split in order to provide an electron to feed into this electron transport chain, or basically to replace the electron that is donated to the electron transport chain in order for the production of NADPH and ATP to proceed. So plants do depend upon water for this reason. But sometimes water is in short supply and plants can become stressed by water. So what is going on with these two pathways? Well, part of what's going on is that generally all plants have these structures called stomata, which kind of look like this, but really tiny. And these stomata are places where plants exchange gases. And the gases they're exchanging are carbon dioxide and and oxygen. And so carbon dioxide typically enters the plant leaf through stomata. These stomata are on the surfaces of leaves. So carbon dioxide will enter the plant leaf through the stomata, and basically oxygen will exit the plant leaf through the stomata. But it's possible for water also to exit the plant leaf through stomata. And this is sort of a natural artifact of the way that plants function. They're taking up water from their roots, but that water is sort of being evaporated as they open their stomata and finds its way out into the atmosphere. Again, if there's a lot of water available, it's not a big deal for plants, but if there's not a lot of water available in the soil, this can start to deplete the amount of water available for plants and make it more difficult for them to effectively conduct photosynthesis. So these two pathways are kind of related to regulating the opening and closing of stomata. When we talk about C4 photosynthesis, we're talking about a situation where carbon capture and the light reaction and Kelvin cycle are really sort of being partitioned into two separate tissues. So carbon capture and fixation's really kind of happening in the mesophyll tissue, as it sort of normally happens during the C3 pathway, but the actual process of the light reaction and the Calvin cycle are instead happening in another tissue we call the bundle sheath. Why is this happening? Well, this kind of allows plants to take in carbon dioxide and then fix it into a four carbon compound and maybe allow the stomata to remain sort of partially closed. And when the stomata becomes partially closed, this kind of drives the concentration of carbon dioxide down and the concentration of oxygen up, which creates some problems for fixing carbon. But in effect, by holding that carbon dioxide as a four carbon compound in the mesophyll, it can help prevent water loss to an extent. And then when conditions are favorable, that four carbon compound can be converted back into carbon dioxide and fed into the Calvin cycle. once the light reaction has happened. And then that carbon dioxide molecule, once it's fixed, can be used to eventually make glucose, just as we saw before. So in effect, we're kind of seeing the process of photosynthesis happening in two separate parts of the plant leaf, in two different kinds of tissue. And it kind of is a strategy that allows the plant to deal with fixing carbon, maybe while the stomata are partially closed, which prevents water loss. CAM photosynthesis is kind of a more extreme version of this theme, and this is basically a situation that allows the light reactions to be performed during the day, but the interception of carbon dioxide really to only be happening at night when conditions are less stressful. So CAM plants typically will open up their stomata at night, take in carbon dioxide, fix it into another four-carbon compound, an organic acid called crassulacean acid, and then the plants will close their stomata as the sunlight comes out. That four-carbon compound will be converted back into carbon dioxide during the day, but the stomata remains closed, so the plant's not losing water, but it's got its carbon dioxide now, and that carbon dioxide could be fed into the Calvin cycle once the light reactions ensue. Once the energy is available from the light reactions during the day, carbon dioxide enters the Calvin cycle, and as we saw before, that would be used to ultimately fix some three carbon compounds that would be used to make sugar. And I think you should notice one of the major differences between CAM and C4 is that C4 is happening really in two different kinds of tissues, whereas in CAM, photosynthesis is happening in the same tissue. It's just happening at different points in time. So to reiterate, C4 is kind of undergoing the process of photosynthesis in two different parts of the leaf, whereas CAM is undergoing photosynthesis at two different times, night and day, but in the same part of the leaf. And again, the important take home here is that these are two different strategies that allow these plants to either partially or completely close their stomata to prevent water loss, but still undergo the normal process of photosynthesis. And then lastly, in terms of variation on the theme of photosynthesis, we can look at how it happens in prokaryotes. And as we know, prokaryotes do not have membrane-bound organelles like chloroplasts, yet some of them still perform photosynthesis, like cyanobacteria, for example. And so how is this working? Well, it's similar. It's a bit simplified, but basically, rather than having these chloroplasts, these prokaryotes have infolded regions of the plasma membrane that sort of function like thylakoids. And so here's kind of an example, a cartoon example of what these look like. And here's an actual electron micrograph photo, or micrograph, I should say, image of what these cleaved thylakoids look like. So despite the fact that there is no chloroplast, there still are thylakoids, and all of the same necessary components that we saw before that are used to carry out photosynthesis are still present. So the light reactions are still happening. The same chemical reaction is still happening where we need both carbon dioxide and water in order to be catalyzed to form sugars and oxygen. But it's just not happening inside of a chloroplast. But we still have, again, the light reactions and the Kelvin cycle and the whole same process that we just saw happening. It's just happening in a different kind of a structure. So this whole idea about photosynthesis and cellular respiration being these two really sort of intricately involved chemical reactions really leads us to thinking about something called the carbon cycle. And the carbon cycle is this process that very effectively moves energy through the biosphere. And if you notice, interestingly enough, the reactions of photosynthesis and cellular respiration do share sort of reciprocal reactants and products. So if we look at the reaction of photosynthesis, carbon dioxide is reacting with water, the reactions being catalyzed by solar energy to produce sugar and oxygen, as we just saw. Of course, a lot of complicated steps going on behind the scenes, but this, in effect, is the way the reaction is proceeding from start to finish. And look at this. The reaction for cellular respiration is the exact opposite, where we have oxygen and glucose reacting in order to form water and carbon dioxide, and the main product of that reaction is chemical energy in the form of ATP. So these are really reciprocal reactions. And this is kind of something that's going on in a cyclical fashion within the biosphere, within ecosystems. Plants are consuming carbon dioxide and water. That reaction's being catalyzed by light energy to form sugar and oxygen. And in turn, animals can consume that oxygen and that sugar. The oxygen allows them to perform cellular respiration, and the sugar gives them energy to make ATP. And in the process, animals are creating carbon dioxide that plants can yet again take up, along with water from the soil, to continue this cycle, this process over and over again. So this is kind of a simplistic picture of what the carbon cycle looks like at the biotic level, at the level of living things. But actually, the carbon cycle is a bit more complicated in how it's moving energy not just through the biosphere, but also through Earth's other spheres, such as the atmosphere and the lithosphere, and perhaps even through the hydrosphere in some cases, if we talk about carbon being absorbed within the oceans. But effectively, we know that carbon dioxide is transient in the atmosphere and it has certain sources. For example, let's say carbon dioxide emitted from volcanoes. And that carbon dioxide could be taken up by plants, which then are either going to be consumed by animals or might decompose into the soil and become things like fossil fuels or oil, petroleum, or natural gas that could be used as fuel. And that fuel, when it's burned, is going to release carbon dioxide that goes back into this carbon dioxide. Or if, say, some of these fuel sources like wood, like biofuels, are being burned, that produces carbon dioxide that goes into the atmosphere and can re-enter the carbon cycle. Or fossil fuels that are burned for transportation are creating carbon dioxide that goes into the atmosphere and can re-enter the carbon cycle as well. So effectively, carbon is being cycled back and forth from generally carbon dioxide into organic forms, typically sugars and other organic molecules, often either decomposed or basically undergoing the process of combustion, by which it is converted back into carbon dioxide and often taken up by plants again. And there's this sort of cyclical thing going on, wherein carbon is basically routinely interacting with the entire geosphere, both in biotic systems and often in abiotic systems. So this is kind of how we think of the carbon cycle. And another interesting aspect of the carbon cycle that I think is worth looking at is the fact that there are global carbon reserves that are composed of both living and decomposed matter. So a lot of global carbon is held up in living plant tissue. Tremendous amounts of it is held in tropical forests. A huge proportion of the Earth's carbon is in tropical forests, somewhat less so in, say, subtropical or temperate forests, and even less so in drier environments. But there is a tremendous amount of carbon held in living organic tissue, mostly in plants, some animals, but mostly in plants. And then a lot of organic carbon is held in dead material. So in the soil where organic molecules have decomposed, where plants have died and decomposed, or other organic matter has decomposed within the soil, we see a lot of carbon captured. And then ancient carbon tends to be held up in gasoline or petroleum reserves, in oil reserves, or again in these sort of pockets within the subsurface of the Earth that we sort of were thinking of here, where carbon-based compounds have decomposed. And these things tend to be like what we think of as fossil carbon, fossil fuels, hence the name. And so natural gas and petroleum really are the remnants of living things that once existed a long, long, long time ago. We're talking about maybe millions of years ago, things that have decomposed and been converted into very, very high energy fuel sources that we tend to rely upon. And we can talk about this concept of carbon sequestration. So sequestration is kind of a fancy word for storage, but the Earth's carbon is generally stored in living tissue, decomposed matter, and in ancient fossil fuels far beneath the ground. And of course, there are inherent challenges associated with relying upon fossil fuels. These things obviously put a lot of carbon dioxide into the atmosphere. Putting a lot of carbon dioxide into the atmosphere is associated with increasing the temperature of the atmosphere, which is associated with not just global warming, but the greenhouse effect, and can have some serious implications in how the greenhouse effect contributes to climate change. And so these are all things to think about, but in effect, the absolute origins of all of this carbon, all these carbon reserves that we see is photosynthesis. If it wasn't for photosynthesis, none of these carbon stores would exist. If it wasn't for photosynthesis, we would not have fossil fuels, and we probably would not have complicated functioning ecosystems like we have today. So that's pretty much it for this topic. In essence, we know that we have this very interesting process by which photosynthetic organisms, typically photoautotrophs, mainly plants and algae, and to a certain extent cyanobacteria, for example, are able to take carbon dioxide and water and light energy can catalyze a reaction to fix that carbon dioxide into a series of organic molecules that are ultimately used to make sugar, and oxygen's kind of a byproduct of it. And those processes happen via basically two steps, two stages, these light-dependent reactions and the Calvin cycle, pretty much universally happening across all photosynthetic organisms, which are kind of complicated pathways. We saw the chemical reaction of photosynthesis, which is simple in nature, but we know behind the scenes there's a lot more going on. And that there's a little bit of variation on the theme. So a lot of plants use this so-called three-compound method, or they are C3 plants, this C3 pathway of performing photosynthesis, but there's also C4 photosynthesis and CAM photosynthesis, which are photosynthetic pathways used by plants in more drought-stressed environments. Of course, we looked at how prokaryotes also perform photosynthesis, but they don't use chloroplast. And of course, we talked about the carbon cycle, which is the sort of ultimate end of how carbon is being cycled through the environment, largely via photosynthetic organisms and the consumers that are eating those photosynthetic organisms and the consumers that are burning these fossil fuels for their energy source. All right, so that's it for this video. I'll see you next time.